Thank you, Nero, and thank you for giving me this opportunity to tell you a little bit about our experience. Unlike the previous talk, I'm going to really focus on the spatial part, and unlike Aviv, who's a sort of basic scientist, I'm a pathologist. I spend some percentage of my time and a larger percentage of my career actually looking at tissue from human patients and making a diagnosis. And that's all about the spatial information. So I want to start with just my disclosures, and I do like to comment here that you can see that I have a number of disclosures, but that importantly, a lot of the work in my lab is now sponsored not necessarily by the NIH. I do have some support from them, but also a lot of the work is supported by biotech and pharma. So this, as a pathologist, this is one of the books that we all use to look at immunohistochemistry. Immunohistochemistry obviously telling us at least some molecular information about the spatial information that we see on the slide. And I show this book because in this book there are 944 pages, of which 9 of which are on quantitative or quantitation or measurement of that immunohistochemistry. So that means from the perspective of the pathology world, about 1% of what we do in molecular measurement is actually measurement. The rest is just molecular opinion or molecular subjective assessment. And so why is this spatial information and measurement of this spatial information so important? So I'd like to show this slide next. This is actually an RNA profile of 2 masterpieces. And on the bottom line, you can see the dark to light line and on the very bottom of each of the pictures. And that shows sort of the intensity. And then the colors are each shown in histograms of how many pixels there are from each of those colors at various intensities shown in the bottom. And so someone in the audience might be able to tell me which is the Monet and which is the Renoir. Because all the information is there. Every single pixel in the actual photos or the actual artwork is there, photos of the artwork in this case, but you just don't have the spatial information. All you're missing is spatial information. So sometimes there's an art history major in the audience, and I thought there might be one here, that says, wow, there's a lot of light blue in the picture on the right. That must be a Monet, because Monet liked light blue. And that's the furthest anyone, and actually a couple people have gotten that one on this quiz. In fact, it is the Monet, and the one on the left is the Renoir, but no one has yet to identify the boating party from the stroll. And that's the critical information that's spatial that we're ignoring when we grind things up or do things in a sort of the fruit smoothie analysis that was given by the previous speaker. Or even if you take and line up all your blueberries and cherries, you still don't get this kind of information. That is the spatial information, where things are with respect to each other, I think is absolutely critical. So that's what I want to talk about. First, I want to just briefly talk about how we measure stuff on slides, very briefly, because that will help us understand what we're doing with spatial information. Then I'm going to reiterate a little bit of what Nero said about our experience with the digital spatial profiling device, how we choose regions of interest, and then how we've done analysis. And then I'll show you our first use of digital spatial profiling as a discovery tool. I'm not going to show you so much the validation as much as the conclusion. The validation is in other papers and other work. And then finally, one of the things that we just did in the last couple weeks is we actually compared the two. That is, looking at using an IO360 panel at 770 RNAs on the Nanostring platform compared to 44 proteins on the Nanostring platform to see is in fact protein equal to RNA. So the first thing is about measuring, and there's 2 ways to really measure. One is to segment and count, which is shown here. And you can segment by defining a threshold. So let's say our threshold is a 15% gray intensity. So then in that case, there's 5 cells that are positive and 5 cells that are negative. And that's segmenting and counting on the basis of a threshold that you've defined. The other way is to measure the total intensity, and that gives you a number, but the number isn't a number of cells, it's about the environment, the whole field of view, in fact. And then you can see on the bottom line, if you measure the total intensity, you got 3 times 35, 2 times 15, and 2 times 5. So you have 145 intensity units per field of view, per square millimeter, per microliter, whatever, however you want to categorize it. And those are sort of 2 intrinsic differences. So the top one is counting. You're going to end up with a counts per square millimeter. And that's one way to do things. And then it's a very different phenomenon to measure as a concentration. The one on the right or the bottom is the concentration, where we actually end up with a mgs per mil type parameter, or something that's expressing not only the number of cells above a threshold, but the true continuous value measuring the intensity of each. I think both are important. And there's some times where you want to count and threshold and count, and other times where you want to measure. But one of the strengths of DSP is measurement, and that's why I wanted to bring this up beforehand, because it allows you to get a true field of view measurement. So 20 years ago, we invented this technology in my lab called Aqua for automated quantitative analysis, and that's what's shown on the left here. And it's actually like a concentration. This is a measurement, and some of the data I'll show you today is measurement by Aqua, which is a per field of view measurement. And we can sort of reduce it to a true concentration. We actually do picograms protein of interest, picograms of target per microgram of total protein. So it's a true concentration. As compared to where another tool we use is actually the threshold and count tool. And in this case, we used either Inform, Halo, or Visiopharm software to do this threshold and counting. Here's an example of the threshold and count, where you can see how many T cells we have, how many cytotoxic T cells, how many B cells, and then how many cancer cells, each with the color code shown in the top rectangle, and the tool which we use to count these on the lower right corner, which is a PerkinElmer Polaris platform. And this allows you to have a different kind of information, but an important kind of information, because not only do we know where, how many of each of those cells there are, but we can also calculate what the distance is between each of those cells. And I'm not going to go through that so much today, because I want to focus more on the region of interest, or the whole concept of measuring in an area. And so to do that, we have the different types of ROI selections. And the one that we're most interested in is the phenotypic one, because the phenotypic one is kind of giving us a region of interest that is defined by a molecular definition. And so we've started to use the term molecular definition of compartments. And that, I think, is very applicable to digital spatial profiling. We did that with Aqua, with quantitative fluorescence, and now we're doing that again with digital spatial profiling. You can also attempt single cell, which is shown on the right, or just have whole regions to measure a whole single spot. But we're going to focus on the one in the middle. And in fact, this is an example of finding regions, which I think will have a lot of value later on. And we're looking at that in terms of user-defined regions, where you can then take a user-defined region and then query a cluster. So you're not only looking at what cells are in the cluster, but what the cells are saying to each other. But I won't going to talk about that. Instead, I want to focus on the molecular compartmentalization, which is not cell counting, but is defining regions of interest on the basis of a molecular parameter. And so specifically, we can use, for example, CD45 to define lymphocytes, or CD68 to define CD68 positive cells like macrophages and other cells. Or we can use more general things like cytokeratin to define a tumor region and things like that. And those are the things that we're going to use as a compartmentalization. And these micromirrors, which are shown on the right and the bottom, allow us to sort of draw, based on the fluorescence pattern, a region of interest that we can actually take. And so that's what's shown here. And you can see that we can draw regions of interest that are defined by the molecular compartment. So we have CD68 as our first compartment that we're going to sip up. Then we're going to have sip up the CD45. Then we're going to sip up the melanocytes in this example, and ultimately the DNA if we have any left. And what we're going to do is, after putting on all those antibodies, and I'll show you those in a minute, we're sipping up just the tags from those antibodies that have been cleaved off, because the micromirror carefully directed the UV light to cleave only in the CD68 compartment, or only in the compartment divided by CD45. So by that way, we're defining a cellular compartment by a molecular tool. And the result of that is shown here. So in this particular one, in the lower right-hand corner, you can see we were using a tissue microarray, and we started with about 60 cases of melanoma. These are all treated melanomas. I think I have that slide next, perhaps. But you can see each of the patient's tissue spots, and then how many counts were sipped up in that amount. And what you can look at is that there's actually a very good replicate between counts. That is, one of the things that you worry about when you're doing this kind of thing is, if I do this again on a second piece of tissue, am I going to get the same result? And the answer on the right shows that actually you get a pretty similar result. So now I want to talk about the actual experiment that we did with with the co-localization. And this shows in higher power the compartments that we defined. You can see the image on the left, which is just a multiplex fluorescence image, but then you can see the compartments that were actually sipped up in the image on the right. And it's hard to really fully appreciate this, especially without a pointer here. But I think that you can see that the cytokeratin, for example, which looks predominantly cytoplasmic and membranous in this appearance, creates a mask under which we can sip up all the counts that are related to the cytokeratin, but isn't perfect. That is, if there's a few lymphocytes in there, they might get sipped up as well. And so we're talking about a regional generalization as opposed to a specific cellular entity that this tool can do. And this is actually then asking the question, what happens if we do them in the opposite order? That is, if we sip up the CK first, and then the CD68, and then the CD45, versus sipping up the CD45, then the CD68 versus the CK, do we get something different? Because if we don't check the order in which we're sipping them up, we might be inaccurate. Or maybe the whole technology is just based on the order. But in fact, as you can see in each one of the cases on the bottom, and then the order 1 versus order 2, there's no difference, or essentially no difference. There are subtle differences between the two box and whisker plots, and each one is from a different patient. But you can see if a patient's high, they generally stay high, and if they're low, they generally stay low. If there's a wide variance in the patient, it stays wide when you sip it up in the opposite order. And so what that means, and it's really critical to me, that we're correctly assigning the molecular compartment, and it's not an artifact of the order in which we've sipped up the tags by the GSP or by the DSP GeoMx. So the first thing coming from the pathology world where we have to make sure we give the right answer for every patient, we also wanted to validate these things against something else that we've already done. So the first thing we did when we, working with Chris Merritt and Sarah Warren at Nanostring, was saying, okay, let's do the Nanostring counts on a TMA that we've already run. And so we used a lung cancer cohort that we'd used many times at Yale that we'd measured with aqua technology or with spatially or with traditional quantitative fluorescence methods, and we compare that. In the upper left-hand side, you can see the CD3 comparison with the Nanostring on the y-axis using I guess this was actually the alpha machine, because this was not our own machine. This was a machine in Seattle. And you can compare that to the aqua scores. And you can see in every case, there's pretty good agreement. In some cases, it gets down pretty low. In fact, you might look at the lower hand, the CD4 in tumor compartment. Well, there shouldn't be CD4 in tumor compartment. CD4 should all be in the stromal compartment, but there is, in fact, some. And in fact, there's heterogeneity. So these slides were a few slides apart. So an R squared of almost 0.4 or 0.38 is, I would consider, an acceptable comparison between these two technologies. So then it's always better, if you can, to compare on outcome, because outcome is the ultimate arbiter. Comparing one test to another just shows the tests are concordant, and it doesn't tell you if they're correct or not. And the correct answer is given by patient outcome. And so what we did in this case is look at CD3, or T cells are known to be a predictor or a prognosticator of bad outcome in lung cancer. And so in this lung cancer cohort, we could see that CD3 in tumor by aqua, both with for overall and for progression-free survival, was quite a good predictor of outcome, and that's shown with significance here. And then we did the same thing with the Nanostring, and we saw the same results. We're slightly not predictive of CD3 in the stroma, but overall the patterns look very much the same, and that increases our confidence that what we're doing and what we're picking out is actually the same result that we would have got with quantitative fluorescence. So we're not just comparing one test to another, but we're showing that both tests are approximately equivalent with respect to outcome. And ultimately, we also wanted to compare some other variables with that we had collected, because the Nanostring lets us look at 44 variables at once, but with quantitative fluorescence, we're looking at only 4 or 5. And so here's some examples of some other variables we looked at. But I like to show this one because it's interesting. In the upper right-hand side, we were looking at EGFR. And as you know, sometimes EGFR can be amplified. And we couldn't detect that because we don't have enough dynamic range. But you can see how the quantitative fluorescence score kind of plateaus. But the Nanostring DSP score keeps going out when we were looking in the CK compartment. And I think that's real. I'm not 100% sure it's real. But I think what we're seeing is that the extended dynamic range of the DSP is shown by the sort of plateauing of the curve where we can't detect any more fluorescence. And we do know from previous studies that for immunohistochemistry, conventional chromogenic immunohistochemistry has a range of about 1 log. And quantitative fluorescence has a range of about 2 logs. And it appears that perhaps the DSP is out at 4 or maybe even 5 logs, which could be very interesting because there are some biology that can't be contained in 2 logs, and especially gene amplification biology. So this is what we did most recently. We've gotten the machine in our own lab because one of the things that I've found in my years as a measuring pathologist is that when people do stuff in someone else's lab, they seem to work better than they do in your own lab. And so the real test for any technology is to bring the technology in and see if you can do it in your lab. And so this is with our beta machine. So I was thrilled when Nanostring honored me by placing a beta machine in our lab, and we got to do it ourselves. And so this is work done by John Zugazagoicha or Swati Gupta, who are both the fellows in my lab that are working on this. And this is how it actually works. So you can't collect the whole thing at one time unless you're going to stay up for about 18 hours. And so over the course of 2 days, they collected each spot. And I think this is interesting because TMA spots, as people who know TMAs know, they're not perfect. And a lot of times, you get something like the little square that you can see on the upper left-hand score, because that's the only part of the TMA we could actually read. And so we couldn't read the whole spot, so we had to read less than that. But we're still okay. We don't want to throw that spot away because it represents a valuable patient information. But this is how it really works, sort of in the real world. And we were actually, in day 1, on plate 1, we collected 24 ROIs from 23 spots. And then in each ROI, we have the 4 compartments, the 4 molecularly defined compartments that I mentioned earlier. And when we do that, we could orthogonally validate, again, in our own lab, our aqua scores with the Nanostring DSP. And you can see that the PD-L1 and the tumor mask was actually quite concordant. And interestingly, the PD-L1 by QIF versus the DSP was not so concordant. The R-squared was 0.18. We'll get back to that in a minute, but I think that's real. I think we have different kinds of compartment measurers and that we're measuring a difficult compartment for quantitative fluorescence to manage. And so with that, I'd like to show you the use of the discovery tool. So here's a melanoma cohort. This is not in our lab now. I'm switching back to work that we did with Sarah and Chris at Nanostring. And this is a cohort, however, that we sent them, this cohort. And this is a cohort that Harriet and Pak Fei accumulated at Yale of 117 patients treated with immune therapy. And about half of them were treated with Ipi-Nivo, and the other half were either treated with Nivo alone or Pembro alone. So either monotherapy or combined therapy. And you can see the distribution of gender and age and stage, et cetera, there. But you see the N is only 59. That's because of the 117 cases, there was only 59 that were large enough that we could make a tissue microarray. And a tissue microarray obviously affords us a lot more assays with each one. So with 59 cases, we used 44 antibodies on the right in one experiment. And we're pretty excited about that when this happened. And this is what we saw. We collected in the 4 compartments that are shown, the S100 MEL17 compartment, that was our tumor compartment, then the CD68 compartment, the CD45 compartment, and then the DAPI compartment. So actually, we got 44 variables or 44 variables in antibodies in 4 compartments, or 132 variables that we've collected on 59 patients. And so here's an example of the collections again, the number of counts collected in the CD68 compartment, then the CD45 compartment, and then the melanocyte compartment. And by the time we got to the DNA compartment, there were basically no counts left because everything was collected in one compartment or another. And on the top, you can see STAT3, and it shows you kind of what you'd expect to see, that you'd see some STAT3 in all of those compartments. On the bottom, you can see CD8, and you can see a sizable amount of CD8 was collected in the CD68 compartment. So at first, it's a little bit of a worry for us, but then we realized actually this is not what we're probably seeing is CD8 molecules that are very close to CD68 molecules. And we shouldn't, and in some sense, that's sort of a pseudo proximity assay. But that's just where it is. That's how well this technology works. It's not truly a defining every cell technology. It's about the environment in which cells live and the compartments we can define molecularly. And so that you can see the CD45 has the most CD8s, which is what you'd expect, and that the melanocytes hardly have any CD8. There's a few that are in interstices of the melanoma that get captured there. And so how do they ultimately look? Well, first thing we looked at was the reproducibility. I'm big on technical analysis, and we wanted the experiments that we did on 1 month to look like the experiments in the next month. And the ones on the bottom, 376.3.2, that stands for tissue microarray 376 block 3 cut 2. And that was a completely different core from the patient than the one on the y-axis, which is 376, that's the same patient cohort, block 1, core 3. And so the fact that we have regressions of beta-2 macroglobulin in the range of 0.82 is pretty amazing. This is even if we're not looking at each individual cell, what we're looking at is highly reproducible in beta-2 microglobulin and HLA-DR and in CD3 to a slightly lesser extent. But look at the PD-L1 and the melanocytes again. Kind of weird. So something might be going on there, and maybe we'll discover that later when we look back at that. And in fact, that's what we found, is that when we looked at where the PD-L1s were and then looked at outcome in those cohorts, we found that the PD-L1 in the melanocytes was in fact not predictive of outcome, but the PD-L1 in the CD68 compartment was statistically significantly associated with response to either combination or single immune therapy. And so I think that's a really interesting phenomenon to think about, since we've seen the field change over the last few years in immune therapy, where more and more people are looking at the quote-unquote immune cells instead of the tumor cells, and this is certainly consistent with that hypothesis. Other things were also interestingly separated into compartments, and we're still understanding this completely, but beta-2M turns out to be predictive in the CD68 compartment, but not in the melanocyte compartment, whereas the opposite is true for HLA-DR, where it's predictive in the melanocyte compartment, but not associated with outcome in the CD68 compartment. And so ultimately, you want to do multivariate analysis to see which one of these things are real, and only 2 survive multivariate analysis, that is beta-2M in the CD45 compartment, and then most importantly, CD68 and PD-L1 in the CD68 compartment. And this we've actually followed that, and this is statistically significant in both univariate and multivariate analysis. But and we followed this up, and I'm not going to show that here, rather just summarize the talk. But in 2 other papers that are now under review, we've found using traditional quantitative fluorescence methods, segment and count type methods, and then doing confocal microscopy, that it is in fact the CD68 compartment or macrophages or perhaps dendritic cells that contain the PD-L1 that's most predictive of outcome in immune therapy, at least in the melanoma, and now also in a second cohort of lung cancer patients treated. So I kind of want to illustrate the discovery potential, because this whole thing came to us as a discovery based on the observations that were first observed on the DSP. So in summary, there was kind of high concordance between DSP, both comparing it to quantitative fluorescence, comparing it to outcome, and comparing it to itself performed on different months. In a single experiment, we found CD8, CD3, beta-2 microglobulin, and macrophages and HLA-DR and melanocytes are all predictive to IO therapy. In the same experiment, we found no predictive value for other interesting markers like Q67, granzyme B, B7H3, B7H4, and VISTA. And finally, this shows the potential to look at many of these things, in fact, 132 variables, protein variables, all at one time with one assay. So this is kind of our now model for potential alternative mechanism of action based on the discovery that we made on the DSP. That is that PD-L1 might be expressed in macrophages or other CD68 positive cells, and that might be the mechanism by which macrophages detect whether or not there's sufficient, whether a tumor is hot or not, or whether there's sufficient inflammatory cells present. And when a T cell comes along expressing PD-1 and binds to PD-L1, it could downregulate signals to attract further T cells, like chemokines CXCL9 and CXCL10. Now we know that those are chemokines that bring T cells to the site, but we don't know that that's decreased yet. This is still just a model that we're working on. But it makes an interesting model because you can see how if you put in an inhibitor like an anti-PD-1, then the macrophage essentially wouldn't know that there were any T cells around because that interaction is inhibited, and that might increase CXCL9, CXCL10, and result in a more hot tumor. And in fact, this is what's been seen and was even shown a little bit by Aviv previously, that after you treat patients, you get a lot more T cells attracted to the site than before you treat patients with this drug. So we're not saying this is the mechanism of action, but it's perhaps an alternative mechanism of action that is now led to a number of other studies that are ongoing to try to prove this is true. The last thing I want to talk about in the last 4 minutes is the central dogma. And that is, we all do all this expression profiling and RNA measurement, and we assume that that's what we're measuring. We're measuring the protein even because it's a lot easier to measure RNA than protein. And as you just saw, you can measure 20,000 of them at the drop of a hat compared to our real difficulty in even measuring more than 4 or 5 proteins at once. And so the question is this really, can RNA be a surrogate for protein measurement? Or are these really regulatory nodes? And sometimes RNA is not equal to protein. Well, this technology gave us the opportunity to ask this question in a rather sophisticated way and a relatively rapid way as well. And what we did is compare the IO360 panel, which many of you might be familiar with, 770 genes on the Nanostring Encounter, or in our case, the Nanostring Max, and do a sort of a traditional mRNA analysis where we took each slide, macro dissected just the melanoma off, and made RNA from that. And then we looked at the RNA profiles, and then compared that to the digital spatial profiling on the same cases. Now, it's not the same slide because we used one slide for the RNA, and then we used the tissue microarray spots for the proteins, which are shown on the right. And because I already explained the compartments, we have 132 protein variables, 770 RNA variables. So obviously, we're not going to be able to compare every RNA, but we can get this. And interestingly, if we look at beta-2 microglobulin in mRNA versus protein, there's a pretty good correlation. And so you can see each of the compartments are shown. First, the S100 is the melanocyte compartment, CD45 compartment, then the CD68 compartment. And in each of the cases, there's actually a pretty good correlation between the RNA that we got, just the bulk RNA that we got from the conventional nanostring methods versus the protein. But look at this one. This is PDL1. The Spearman's correlation coefficient ranges from 0.19 all the way down to 0.04 in the CD44 compartment. So RNA really doesn't equal protein. And in fact, if we look at all the markers, we can kind of summarize them with using an R-squared or using a Spearman rank on the right. You can see the molecules that correlate and the molecules that don't correlate. So there's a really strong correlation for CD8, CD56, beta-2 microglobulin, B7H3, and HLA-DR. And so for those molecules, you're probably fine looking at the RNA and inferring that those proteins are expressed in those cells. But the opposite is true is for STAT3 or for granzyme B. I'm not sure about B7H4, but all of those things actually seem to be the opposite. There's an anti-correlation or a weak anti-correlation compared to the RNA, between the RNA and protein. So this is kind of hot off the press data. We still have a lot more analysis to do on this. But I think that this at least illustrates something that we've seen before, and that is that RNA doesn't equal protein, but protein does equal function. Protein and post-translational modifications that occur to protein really are how functions occur. And it's one of the reasons I'm enthusiastic about new protein monitoring tools and the ability to high-plex protein is the fact that we can perhaps more closely probe protein with these tools than we were able to do by just looking at bulk RNA or even single-cell sequencing. So with that, I'd like to just quickly flash up all the people in my lab who are involved in this work and who actually that I get to talk about and show their pictures here. And I guess I can take questions if we have time. Thank you.
If you have questions in the audience, just wait for the microphone so you can speak into that. While we wait, I had a question for you, David, which is on the last part that you were talking about between correlation in RNA and protein, and thinking about areas where you see high levels of correlation where you don't. Is there a way for us to a priori know the regulation of these targets to be able to predict where the correlation might be high and low? Because when we think about protein targets, does that inform us on where protein information is going to be more valuable, where the RNA might serve as a surrogate?
That's a good question. That is, when do we need the protein and when do we need the RNA, so to speak? And I don't think we know yet. I mean, if you start to look at, so from my perspective, it's always how can we take this to the clinic and what can we use this for in the clinic? And clearly, one of the big clinical needs right now is the ability to predict who's going to respond to immunotherapy, because that's a drug that's not a non-toxic drug. Yes, some patients do very well with it and others die because they took the drug. So you want to be able to have some diagnostic test that picks out which patients need the drug and don't. And in fact, all we have right now is immunohistochemistry. So that's an example where the protein might be really important because we know the target. But other examples, RNA signatures have become more important. For example, in breast cancer, when we're deciding to give someone endocrine therapy plus minus chemotherapy, we use RNA platforms, either the MammaPrint or the Prosigna 50, or the most common is the Oncotype, where we're just using an RNA platform, just ground up RNA. So I think that both tools can be used. I'm not sure how we tell which is which, other than doing the kind of stuff that we're doing, is testing them out, and I think actually both, the platforms where you look at single cell sequencing and find out which RNAs are really important, and then where those are localized potentially, or just finding out where they are, which RNAs are important, you can build signatures that can potentially predict response to immunotherapy. In fact, we know the Ayers work that was published describing response to pembrolizumab based on an 18-gene signature done on the Nanostring platform. There's also a 3-gene signature done on the Cobas platform, I believe, from Roche. And then we have our own RNA signature. So there's an example where an RNA signature can get you there. But I would argue the spatial information with the proteins is likely ultimately to be more specific. People criticize the protein information. They say, well, you've got to be wrong because IHC for PD-L1 has been so bad. Well, that's a function of how it's done. That's not a function of the protein information. It's a function of poor quality assays, non-reproducible and non-harmonized assays, then being read by a pathologist, expecting us as pathologists to read in ranges we can't read consistently. We do fine in the high ranges, but in the low ranges we don't do so well. And the result of that is the assay doesn't look so good, but not because the protein is not the way to assay the phenomenon, but because the assay itself is failing.
Any questions from the audience?
Excellent talk, thank you, David. So talking about the quality, so certainly using a cocktail of 44 antibodies for staining is probably challenging. So what is your recommendations? I mean, do you trust it? So because it is, it should be optimized panel, and how to optimize it.
Right, so that's a great question.
For this particular tissue and for TMA.
Right, I'm a real stickler on antibody validation. I've published extensively on antibody validation, on how non-validated antibodies can also lead you astray as well as provide good information. And so when Nanostring was putting together this panel, I was really challenging them on making sure that they were using validated antibodies. And even though I didn't show you the antibodies at Nanostring's request, those are antibodies that I'm familiar with, and many of which I've already validated in my lab that are in that panel. So I have a high degree of confidence that the antibodies themselves are binding to the right protein, that it's not an off-target effect or like a polyclonal where you have a lot of binding of non-specific binding. The second question you ask is more complicated because this is the first time I've mixed 44 antibodies together, and there could be steric hindrance types effects. They're like maybe the CD3 is getting in the way of the CD8 because they're both on cytotoxic T cells. And I don't know the answer to that question. I think that's a valid question and something that we need to look at going forward because I want to mix 100 antibodies together or maybe even someday 1,000 antibodies together and see and do the same kind of multiplexing within specific regions and compartments. And so, experiments may need to be done to prove that the mixed antibodies resolve in the same way that single antibodies do. We've certainly done that with multiplexing. When we do high-fold multiplexing, we compare the multiplex versus the uniplex for that antibody, and we do always do a regression. We haven't had the opportunity to do that yet, to look at individual molecules versus mixing all the antibodies together. So it's a good question.
Great. Thank you so much, David.